So, hello everyone, this is Rishabh, and today we are going to study about hematogenesis and hematology. So, if we break up the name, both has heme, right? What does heme means? Heme means something related to blood, and genesis means development, while the tology means study. So, hematogenesis is concerned with the development of the blood and the vascular system development, while hematology is the study of the blood itself. So, if I talk about the development of the blood vascular system, that begins at day 17th or second week of embryonic development. So, at the 17th day in the embryo, inside the yolk sac, mesodermal cells comes and they get arranged in form of clusters. They are arranged in form of clusters. And what is the name of the clusters? These are known as blood island, as I have also told you in the cardiovascular system. These blood island have those cluster of cells, those mesodermal cells which form the cells forming clusters. So what is the name of the cells which are forming the cluster? They are nothing but known as hemangioblasts. It means mesoderm came. It formed this hemangioblast cells. Hemangioblast cells form this cluster, and this cluster is known as blood island, right? Good. So, after we have made this cluster, what will happen is that this cluster will be differentiated. So, if this is our blood island, this hemangioblast cells, which are present at the periphery, will give rise to our blood vessels. This will give rise to blood vessel, and those hemangioblasts which are present in the center, this will give rise to blood cell. Don't worry, I will make you remember it. Suppose this is a blood vessel and I just took a section. What you will observe? You will have the blood vessel at the periphery, which is developed from the peripheral hemangioblast. And in the center of blood vessels, what do you observe? You observe cells. So these are developed from what? Central hemangioblast. See, if you see the name itself, hemangioblast. Hem means blood, angio means blood vessels, and blast means cells which are capable of division. So these cells will divide and it will form the blood vessels and they will form the cells of the blood. Our curriculum is primarily focused on to the development of the blood, or you can say hematogenesis. Let's see. You have a multipotent cell. This is the similar cell which was the central hemangioblast. This central hemangioblast is a multipotent. As the name says, multi means many, potent means potential. It means this cell can divide to give rise to many kind of cells. What many kind of cells? We will see. So see, this multipotent cell or the central hemangioblast will divide to form myeloid stem cell and lymphoid stem cell. Myeloid and lymphoid stem cell This lymphoid stem cell will ultimately form your lymphocytes, while myeloid stem cell will form your non-lymphocytes. For example, if you need RBC or the erythrocyte, this myeloid stem cell will differentiate into erythroblast, it will form reticulocyte, and from reticulocyte this will form your erythrocyte. Again, if you need platelet, myeloid stem cell will differentiate into megakaryoblast. Megakaryoblast will differentiate into megakaryocyte, and megakaryocyte will further form your platelet. Right? Again, if you know, and like I will tell you about it later, your lymphocytes are divided into two parts. Sorry, not lymphocytes. Your leukocytes is divided into two divisions. Lymphocytes plus monocytes as agranulocytes. Agranulocytes means they don't have lysosomal rich granule. What does it mean? It means lysosome enzyme वाले granules वाले vacuoles इनमें नहीं होंगे. But those two which have it, for example, neutrophils, eosinophils, basophils, they are known as granulocytes. So you have to make these granulocytes, for example, neutrophils, basophils, eosinophils, your myeloid stem cell will differentiate into myeloblast. Myeloblast will directly develop into eosinophil and basophil, but can form the band myeloblast. Band myeloblast is where neutrophils has many lobes. These lobes are fused in this myeloblast. Again, if you have to make the monocytes, monocytes are present in the blood, but if that is present in connective tissue, we say it macrophage. They are phagocytic cells. It's just, the relative term. For example, lungs, this will be known as dust cells. In the liver, known as Kupffer cells. In the spleen, reticuloendothelial cell. So this monocyte, if we have to make, our myeloid stem cell will differentiate into monoblast, and this monoblast will form our monocyte. Similarly, we have our lymphoid stem cell. Lymphoid stem cell will form our lymphocytes. For example, how many lymphocytes we have? We have T lymphocytes, we have B lymphocytes, and we have non-killer cells or natural killer cells. We have T lymphocytes, B lymphocytes, natural killer cells. If we have to make any of them, we will have to make our lymphoblast. So our lymphoid stem cell will differentiate into lymphoblast. It will form our B cells or T cell and our natural killer cell. You might have this in your mind, भैया T lymphocytes के भी further type होते हैं तो वो कैसे बनते हैं? See dear, you have T lymphocyte. T lymphocyte is an inactive lymphocyte. When this comes in contact with any pathogen or any foreign object, this gets activated, and upon activation it differentiates into its various form. जिसके बारे में मैं डिस्कस करूंगा, don't worry. So a quick revision. Second week, mesodermal cells came. They formed a cluster of your hemangioblast. Hemangioblast were arranged. This is the cluster of hemangioblast, and the cluster is known as blood island. In that, the peripheral cells will give rise to blood vessels. The central cells will give rise to blood cells. Got it? Great. Now, mark my word. We just completed the blood formation in the yolk sac. But yolk sac is not just the part जहाँ पे blood बनता है. You have your kidney, you have your liver, you have your spleen. There are lots of structure where blood formation takes place during the embryonic development. For example, earlier in the fourth week of development, hematopoietic cells will colonize your liver. This happens just after, like just after the erythropoietic erythropoiesis in the yolk sac. It was on the 17th day, and this will begin on 23rd day. So if someone asks you the place where erythropoiesis occur in the adult, you can say bone marrow, but in uh, during embryonic development, not just our bone marrow, yolk sac, our liver, in our spleen, bone marrow, of course. So within third week of gestation or the 17th day, the yolk sac begins to show hematopoietic activity, I told you. Early in the fourth week or day number 23, first wave of hematopoietic cells will colonize your liver, and that will be coinciding or that will be simultaneous with the tapering off of yolk sac blood cell activity. Again, you will have on to the day number 27 or week 4 abdominal arterial clusters. Now, these clusters will be active in the abdomen. Later, they will colonize your liver on day number 30 or week 4. See, you don't have to study it in so much detail. Even if you know like कौन से organ में हो रहा था development, that is more than enough. Okay. Now we have colonized our yolk sac, our liver, our spleen. What about the bone marrow? Bone marrow is colonized in the 11th week of gestation. This is your red bone marrow. Don't worry about this. This is your yellow bone marrow. Initially, during the child, red bone marrow is present in this part also. But as the child progresses to grow, the red bone marrow is converted into yellow bone marrow, and this remains as just fat. So, this was erythropoiesis, where we formed all those cells. I will talk about it, how we make helper T cells, killer T cells, suppressor T cells, memory T cells, plasma cells, and everything. Don't worry, classification हम देखते हैं. We are just talking about the development right now. Now let's talk about the lymphoid organs. So, what are lymphoid organs? Do you know? Lymphoid organs are the organs which either is the place of formation of lymphocytes or it provides the place for action. For example, suppose your lymphocyte is a Beyblade. Just give me a minute. Suppose your lymphocyte is a Beyblade, right? So this was made where? This was made in the factory, and you will use it where? You will use at your home sweet home, right? So you will play here at home. So the place where lymphocytes are made or the factory of lymphocytes are primary lymphoid organ, while the place where they perform the action and the action of lymphocytes is phagocytosis or inactivating the pathogen. The place is known as secondary lymphoid organ. So just a minute. The place of formation is primary lymphoid organ. The place of action is secondary lymphoid organ. Primary lymphoid organ means where the lymphocytes will be formed. How many lymphocytes do we have? We have T lymphocytes and we have B lymphocytes. They will be formed in the primary lymphoid organs. And how many primary lymphoid organs we have? Among many, we have majorly in adult, bone marrow, and thymus. Here, production of lymphocytes will take place. The action of lymphocyte will take place in the secondary lymphoid organ. For example, spleen. For example, tonsils. For example, our appendix, our lymph nodes. Intestine has its own lymphoid organ, which is Beer's patches of intestine. So these are the secondary lymphoid organ. Your lymphocytes will be produced here and they will perform their phagocytic activity or whatever kind of activity in the secondary lymphoid organ. It means T lymphocytes and B lymphocytes में बनेंगे, spleen, tonsils, appendix यहाँ पे वो fight करेंगे. Now remove them all. Now let's talk a bit about our cells, right? Let me just give me a minute. We will talk about the cell. Let's first discuss about our thymus. So just now I told you about thymus is a primary lymphoid organ. It means here our cells will be forming here and they will be maturing. If I talk about the structure of your thymus, just mark my words, I won't be writing a lot of them. This is divided into lobules. Okay. How? Suppose this is your thymus. Thymus will have septum of connective tissue which will make its capsule. Now this capsule will go inside and it will divide it into lobes. Now these lobes will further go inside and divide it into the lobules. Just a rough interpretation. So your thymus is divided into thymic lobules, which are separated by what? These are separated by the septum formed by the connective tissue. Now each lobule is divided into peripheral cortex and the central medulla. You know, everything has its cortex and its medulla, right? Now, if I talk about the thymic cortex, just give me a minute. If I talk about the thymic cortex, then it is differentiated into three layers. First layer is your superficial layer. Superficial layer is most superficial part, and that will be superficial subscapular cells forming a squamous sheath and blood thymic barrier. See, this is a lymphoid organ and यहाँ पे immature blood cells होंगे or you can say immature lymphocytes. If the blood has access to this, ब्लड को तो नॉर्मल lymphocytes पता है ना, immature lymphocytes के बारे में पता नहीं है. So if the blood comes in the contact with these immature lymphocytes, your blood will think this is a foreign object or any pathogen, and this will try to destroy it. So to prevent that, what we do is we make some sort of barrier. This is like a barrier. So in this barrier, all the essential nutrient will pass from the blood to the tissue, thymic tissue, and from the thymic tissue, all non-essential elements will pass, and there will be no direct contact of your blood and the thymus, and that is known as blood thymic barrier. These are made by what? These are made by your, just let me change this. You will have squamous cells. They will be resting upon their basement membrane. This is your endothelium or the blood vessel wall. Okay, they are just like this. And then you will have simple squamous epithelium cells just beneath your cortical connective tissue septum. They will have this kind of tight junctions, and with this tight junction, only essential stuff will be passing through it by transmembrane or whatever kind of transportation. So they won't be able to, blood won't penetrate, only the stuff transfer take place by diffusion, osmosis, or whichever way. Not a thing to worry about here. Just remember, blood thymic barrier will be formed by squamous sheath cells and blood thymic, sorry, squamous sheath cells and your these endothelial cells. जहाँ पे endothelial cells will only line, just simple squamous epithelium of the beneath connective tissue will form those membrane. If you don't understand this part, just let me know. मैं तुम्हें ये फिर से बता दूंगा in our next lesson. So, this was about our thymus superficial layer. The second layer will be stellate. Second is our middle layer. This will have stellate, thymic, epithelial, and cytoreticular cells. Then we will have our third layer, which will have inner layer will have squamous, cortical, thymic, epithelial cells, and they will form what? They will form the cortico-medullary boundary. It means what? Suppose this was your thymic lobule, I told you. In the thymic lobule, you had peripheral cortex and you had central medulla. So I told you you have three layers. Layer 1, outer layer, layer 2, middle layer, and layer 3. This layer will be forming a boundary between your cortex and your medulla. So just cortico-medullary barrier, nothing so much important. Now when we come to the medulla of our thymus, this will be very important. Just listen to me. A second layer of squamous epithelial cells and cytoreticulum will be present. Here the thymic epithelial cells, epithelium, so simple squamous epithelium resting onto the basement membrane, जिसके नीचे lamina propria or areolar connective tissue will be present. These epithelial cells will be concreted like a form of, you can say, clustered, and they will form Hassel's corpuscle. Besides these cells, you will have some nurse cells to take care of it. Okay, how they will take care of it? They will be educating it. Educating it means there will be like in physiology you will study about negative selection. Here nurses will check upon the cells which are working fine and properly and which are not. They will be discarded out of the system, and one out of 100,000 just makes out of it. So if we talk about the function of our thymus at the end, here maturation, education, and formation of lymphocytes will take place. This is a histological image. You can see these are what? This is connective tissue capsule. These are the septums coming from the capsule. They are forming these lobules. Lobules has what? These have cortex here. It will have three layers, and this is your medulla. Okay, and what you can see here, these are your blood vessels. Let me just highlight it here. These are your blood vessels. This is how you will see your thymus. So this was your thymus. Now let me just talk to you about this part. You have blood, right? If you blood has 55% plasma and 45% formed elements, not cells, because some are not the cells. For example, white blood cells are not cells, they are corpuscles. 55% plasma and 45% formed elements. Among these 45%, you will have your RBC, WBC, and your platelets, right? Now see, RBC, WBC, and platelets are very different kind of cells. For example, RBC is primarily responsible for gaseous exchange, transport of gases for the gaseous exchange. WBC is for fighting the infection, and platelets are to form platelet plug formation to prevent blood spilling, or to prevent hemorrhages. Their lifespan is also different. For example, RBC will have 120 days, WBC's few weeks, and similar for the platelets. And just because of the lifetime, for example, 120 days, suppose a blood cell was formed, one RBC formed on 1st January. This will die into somewhere around the end of April. Suppose 100 million RBC were formed and they will die in the April. So ये तो मर गए, अब तो हमें new cells चाहिए. This is the reason why I taught you about this part. Hematogenesis. This begins during the embryonic development in different organs, but later this will continue in rest of the organs. For example, your uh, bone marrow. In the bone marrow, I will also tell you about myeloid tissue, yellow bone marrow, and rest of the stuff. So don't worry. So yes. So because cells are going to die, we need to make new cells to fill their emptiness or fill their loss. So this was about our blood. Now, RBC doesn't have its type. Platelets doesn't have its types. Blood, but WBC, of course. White blood corpuscles. They are primarily responsible for what? Phagocytosis, making antibodies, and other stuff. So if someone is doing phagocytosis, it means उनके पास क्या होगा? They will have lysosomes or lysosomal rich vacuoles, right? That will be chiefly seen here. And if I talk about white blood corpuscles, you have two kind of cells which are agranulocyte and these are granulocytes. See, agranulocytes comprises of lymphocytes plus monocytes. Granulocytes comprises of neutrophils, eosinophils, and your basophils, right? Lymphocytes further comprises of T lymphocytes and your B lymphocytes. Let's start with our lymphocytes. Tell me, why do we have T and B lymphocytes? The reason is both T and B lymphocytes develops where? Into the thymus, but maturation of T lymphocyte takes place in the thymus. That's why thymus का T है इनके पास में, and those lymphocytes which will mature into the bone marrow, this will be B lymphocytes. Or you can say, suppose you had 10 lymphocytes, suppose 4 lymphocytes, they all formed into the thymus. Some matured, अगर ये mature thymus में ही हो जाएंगे, so T lymphocytes. And if they did not mature and they were migrated to the bone marrow, those lymphocytes will be B lymphocytes. Actually, the B doesn't mean bone marrow, it means bursa of fabrica. That is a lymphoid tissue found in the birds और उनमें ये discover किए गए थे. But this is somewhat, analogous as yes, analogous to our bone marrow. So we continue to use this term, bursa of fabrical lymphatics or B lymphocytes. So if they mature in thymus, T lymphocyte; if they mature in bone marrow, B lymphocytes. Now, in the blood, T lymphocytes and B lymphocytes remains inactive. When they come in contact with any pathogen, they get activated. What did I say? Normally, they remain inactive, but when they come in the contact with any of your phagocytic pathogens, not phagocytes, pathogens, they will become active. So let's see our T lymphocyte. They are the most abundant T cell. They will identify, not just, they will identify and they will destroy it. Okay, but when they will identify, upon identification, they will get activated, and upon activation, they will divide into helper T lymphocytes, killer T lymphocytes, suppressor T lymphocytes, and your memory T lymphocytes. Helper T lymphocytes will be most abundant. They will, they will identify the foreign cells or body or particle, and they will inactivate or sensitize them. And the sensitized helper T cells produces lymphokinins. Lymphokinin will stimulate other lymphocytes that will, you know, stimulate B lymphocytes also, and they will attract your macrophages for phagocytosis. Then is your killer T cells or the killer T lymphocytes, also known as CD8 cells and T8 cells, and they are known as CD4 cells or T4 cells. Killer T lymphocytes will attack and destroy any invading microbe. This can be cancer cells also. This can be any microbe or any infected cells also. They will secrete perforins. Perforins. See, perforins का मतलब क्या है? Pore creators. So they will secrete pore creating chemicals which will create the pores in, for example, bacteria. And if there is a pore in bacterial cell membrane, content will leak out and some content will go inside and the bacteria will die. Then there is the suppressor T lymphocyte. See, if your lymphatic system is very active, it can damage your own body. So you have some cells known as suppressor T lymphocyte, which will suppress your immune system and prevent it from attacking its own cell. Next is your memory T lymphocytes, which will like it will retain this information, or I can say they are previously sensitized or newly sensitized. Sensitized means interacted, and they will retain this information. Information about what? They will retain information about the pathogens. So this was T lymphocytes formed in the thymus, also matured in the thymus. Next is our B lymphocytes. B lymphocytes are also inactive. That is why when you see a B lymphocytes, their nucleus appears dark blue. Why? Because they are inactive and inactive chromatin is dark, known as heterochromatin. So you will see just cells with a dark blue nuclei. So, but when it is sensitized or it is activated by the T lymphocytes, or when it itself find any pathogen, it divides into effective B cells और जिसे हम प्लाज्मा सेल कहते हैं और memory B cells or memory B lymphocytes. Plasma cells, see, they were inactive and now these are active. So let's see. I told you a concept. If something is inactive, उनका nuclei ऐसा होगा, right? Heterochromatin. Now, since they have become active, it means their nuclei will also be active, their chromatin will also be active, and chromatin, which is active, is known as euchromatin. So, in the nuclei, you will see a combination of hetero and euchromatin, and because of this, it appears like a cartwheel shape. That is why we have said this. The nuclei of the plasma cells appears as our cartwheel shape. Now you got that. It is this cell which will form the immunoglobulins or also known as antibodies. If you want to know the classification of antibodies, they are immunoglobulin gamma, alpha, delta, epsilon, and mu. They can produce almost, like 2000 antibody per second. And tell me one thing, if something is, मैं इसके बारे में अभी बता दूं क्या? Okay, suppose something is producing antibodies and antibodies are what? Gamma globulins. So if I want to make a cell, will it look like this? Yes. Will it have nuclei? Yes. Nuclei will have cartwheel shape. Yes. Just give me a minute. Nuclei will have cartwheel shape. Besides, if something is producing large amount of this antibodies, then antibodies कहाँ जाते हैं? Cell के बाहर. And if some protein has to go outside of the cell, they need to be packaged. And packaging is done by what? Packaging is done by our Golgi complex or the Golgi bodies. And Golgi body is located where? This will be located in the perinuclear region, and this is why this is known as perinuclear HOF. when you see this in the histological slide, this appears as perinuclear HOF. Let me show you. See, if I show you, can you see this? Just a minute. This is your nuclei pushed at one pole. The cell is oval, right? You can see light band here. These are the combination of heteroendiochromatin. And just now I told you perinuclear HOF. Can you see this white color region? This is nothing but your Golgi complex. This will have all of those antibodies. So this will pack the antibodies so that it can send it out. This is, blood image, and this is, uh, what do we say? Biopsy image. The image of cells in the blood and the image of cells in the biopsy is quite different. For example, you can see it here, you can see it here, you can see it here. Don't worry, मैं इसके बारे में बताऊंगा. So we talked about B lymphocytes and the T lymphocytes. In the B lymphocytes, we had our memory B cells. They will store the information about the microbe, and they are stored in where? These cells are stored in spleen and lymph nodes. So this is our T lymphocytes and B lymphocytes, or we can say we studied about our lymphocytes. Now monocytes, they are the macrophages. They will have kidney-shaped nuclei, and these are nothing but the policemen of the connective tissue. They are known as macrophages in the tissue, monocytes in the blood. Now we have granulocytes. What does it mean saying granulocytes? Granulocytes means they have lysosomal rich granules. So they will have lysosomal granule-like structure in their cytoplasm. Let me show you here. So we had neutrophils, eosinophils, and our basophils. मैं basophils भी दिखाता हूँ. See, first of all, it would be better if I talk about the cell type and not the photographs. I will discuss those photos also, so don't worry. Neutrophils, eosinophils, and basophils. These are nothing but our these are nothing but our granulocytes. It means they are rich in lysosomal granules, and they will have variations in their shape and their function. Guys, one more thing. Here we divided, the WBC into agranulocytes and granulocytes, and we had lymphocytes into T lymphocytes, B lymphocyte, and we have non-killer cells, NKC, or the natural killer cells. They are also there. They are non-B, non-T lymphocytes, and they are wandering macrophages. They will perform the phagocytosis, and they don't need specially to come out the blood vessels. They are present there. So we have neutrophils. First of all, most abundant. They will stain with both acidic plus basic dye. So you can see they are also acidophil or basophil. बेसोफिल का मतलब बेसिक डाई होता है. Acidophil means acid, sorry, not basic dye, structure which are stained with basic dye. So we have neutrophils. Let me just write with the small alphabets. So Neutrophils most abundant around 54%. Okay, they will stain with both acidic plus basic dye. Okay, if we see the cell, the nucleus will be multiple lobed. That is why this is also known as polymorphonuclear cells. Okay, these are also uh, you know phagocytes. Okay, and another name of neutrophils is micropolicemen. We had macrophages as macro. These are micropolicemen, and these are the smallest WBC. I will show you the image here. Let's see. This is your neutrophil. You must be asking, भैया यहाँ दो image here. Left side we have blood smear, right side we have biopsy. Biopsy is nothing but taking sections in a very thin image. For example, this is a sample of tissue. We took many sections, and now each section is just a micron thick. And when we will observe this under the slide, this is nothing but called a biopsy. So this is your neutrophils. You can see multiple lobed. This is multiple lobed in blood smear. But when you see this into the biopsy, this is not multiple lobed. This will appear as bent shaped. You see, I will use the color red. You can see here, this one will be bent shape, bent shape. This will be bent shape, bent shape, bent shape. So here the nuclei is not polymorphonuclear, but is bent shape. Why I wanna say is that don't get confused or don't get sad कि यार भैया histology नहीं समझ आ रहा. Here the thing is, histological images are quite different when you see those in the smear, as in the case of blood, or when you see the biopsy. So this was your neutrophil. Next is our eosinophil. Eosinophils are also known as acidophils as they get stained by the acidic stain. Nucleus will be bilobed. They will resist infection by increasing their number. They are active during any parasitic infection. For example, if there is Schistosoma hematobium or blood fluke, in that they will increase their number. They will secrete histamine, and histamines are what? Vasodilator. So they will dilate the blood vessel so that more blood can come here. Next we have basophils. As the name say, basophil, they are stained by basic dye. They are least abundant, or you can say their count is very low. For example, 0.5 to 1% among the total lymphocyte count will be total leukocyte count will be there. Nucleus is S-shaped and stained with basic dye, and they will secrete heparin or heparin sulfate. They will secrete histamine and serotonin. Heparin will be anti-blood coagulant. Histamine will be vasodilator, and serotonin will be vasoconstrictor. It's not कि ये एक साथ सारी चीजों को produce करेगा. No, as the reaction produce, their concentration will change. Okay, I have changed the color for your better understanding. As the reaction will proceed, they will be changing the concentration. And with this, our lymphocyte is also completed. If I want to show you here, neutrophils I had told you, see, eosinophil. In the eosinophil blood smear, let me tell you one thing. In the biopsy, you won't be able to see the cell membrane. cell membrane will not be visible in any biopsy image. So eosinophils or your acidophils, you will see these pink dots. These are nothing but the lysosomal rich granules. That is why granulocytes. Again, here you can see those dot lysosomal rich granules. Again, this one is your lymphocyte, so no granule. And in the plasma cells, no. Here you can see this is your eosinophils, and eosinophils are bilobe nuclei. So yes, you will be seeing those bilobe nuclei, right? Here they will be appeared as bent or kink-shaped nuclei. With this, we have one more left: macrophages. Macrophage. Macro means large, phase means phagocytic cell. A large phagocytic cell. See, if there is a cell which has to devour another cell, if the cell is dead, first of all, protein will be degenerated. Okay, protein will be degenerated inside the cell, because अगर protein functional होता तो cell death ही नहीं होती. Protein will be degenerated, or you can say not a degenerated, denatured. Then they will have nucleic acid. Nucleic acid will also undergo pycnosis, pyridoxis, pyrolysis. So that will, your nuclear membrane, nucleus, nuclear content will be destroyed all. So what you have left is just the cytoplasm and your cell membrane, right? Cell membrane is made up of what? Phospholipid. And I have told you, cytoplasm appears pink, nuclei appears blue, lipids colorless, fat colorless. Lipid and fat are same but different in the biochemistry you have studied. Let's leave it lipid. Glycogen colorless, mucus colorless. So if I told you your protein is degenerated, nucleic acid is also degenerated, what is left? Phospholipid. Phospholipid is colorless. Let me show you your macrophage. This is your macrophage. Can you see this entire part? This is nothing but the vacuole in which there was our phagocyted cell. In this part you can see this is the large cell with just the central nuclei, and what you see, these are just the vacuoles with the phagocyted part of the cells, or you can say the phospholipids. And with this we have completed our hematology and hematopoiesis.